Okay, thank you, Philomena, for your introduction, and also I would like to thank Cephalochordate for the kind invitation for me to come here and collaborate and also work with you guys on some amphioxus research. So today I will introduce some of our recent study. Is it okay?
Yes,
okay.
We better fix because otherwise this will happen again. So you have less wobbling. We put on this part here. It's a little bit loose. Let's try like this. Let's hope it doesn't move.
Okay, thank you, Ina. Okay, so today I will introduce some of our recent study and also some previous study along the line of understanding how vertebrate character evolved. So my main focus, so I will, this is the outline of my talk. So I will first introduce some of the stories that I have been doing to understand how the vertebrate novel characters, including like the neural crest, develop, and how this genetic regulatory network of the neural crest development can be evolved in the chordate lineage. And also I will talk about is that okay? Okay, I will talk about the developmental mechanism of the peripheral nerve system in amphioxus and its implication to how the chordate nervous system evolved. And also I will talk about some of our recent study on the evolutionary origin of the skeletal tissue evolution importance. So in this morning, I've already gave a short introduction to the graduate students here about why we use amphioxus and introduce a little bit about the amphioxus system and its embryology. So I will skip this part and go straight into why my topic today. So this is just a simplified phylogenetic tree of our current understanding of the deuterostome animal. So within the deuterostome animals, there are three major phylums, including the echinoderm, hemichordate, and the chordates. And within the chordate lineage, there are three major clades, including the cephalochordate, amphioxus, tunicates, and vertebrates. And all these chordates have several key characters, including the dorsal nerve core, nodal core, segmented somites, and pharyngeal gill slits. But compared to the vertebrates, there are several novel characters were evolved during the evolution of vertebrates. This novel character, including that the vertebrate genome have undergone at least two rounds of whole genome duplication. So compared to the invertebrate chordate or even to the invertebrate deuterostomes, vertebrate animals usually have more regulatory genes due to this whole genome duplication. And also during the evolution, vertebrate evolved a much elaborated head region with many specific sensory organs that enable these animals can become predators. And also, and these novel characters, this elaborate head structure, are mostly come from two major embryonic tissues, namely the neural crest and placos. And also, vertebrate animals evolve internal skeletal tissue that composed by minorized cartilage and bones. So my purpose of using amphioxus to study for my research, I'm mainly trying to understand how these novel characters can be evolved by comparing the developmental mechanism of vertebrates and cephalochordates. So the first topic I will touch upon is about the evolution of the nervous system. So when we compare the nervous system in vertebrates to invertebrate animal, And we can categorize the nervous system into two major parts. One is the central nervous system, which include the brain, the spinal cord, and the other part we can roughly say them categorize as the peripheral nervous system. And if we look at the central nervous system between vertebrate and invertebrate animal, they are right now we know they are induced or developed by very similar molecular mechanism. So this I just show you a later view of the developing amphioxus embryo using SOX-B1 as a neural marker. As I told you, chordates have their central nervous system on their dorsal side, and this SOX-B1 gene can label the developing central nervous system on the dorsal side of this embryo. And now we know that the inhibition of BMP signaling pathway is very important for chordates and even for other invertebrate animals to develop their central nervous system. And we can use phospho-SMAD 158 staining as the readout of the BMP signaling level. We can see that in the ventral part of this animal, we can detect a very high BMP signaling level, but on the other side, the dorsal side, the BMP signaling level need to be repressed. And this repression is achieved by the expression of the BMP antagonist, chordate, on the dorsal side. And on this other side, we can see some other genes expressed in the ventral part like this HEX gene that enables the ventral structure to be developed. And this process is also is very similar in vertebrate. And in vertebrate situation, it has been known that if we disturb the BMP signaling during the dorsal-ventral patterning, will disturb the overall dorsal-ventral structure and also disturb the formation of the neural tube. And it's similar that in amphioxus, if we disturb BMP signaling by, in this case, elevating BMP signaling by adding more BMP protein, we can elevate the BMP signaling level all around the entire animal. And by this way, you disrupt the formation of central nervous system and ventralize the entire animal. And the dorsal structure becomes disappear from this animal. And this is very similar to other the central nervous system developmental mechanism in other animals. So the homology of central nervous system of the amphioxus to other animals is very clear. But how about the peripheral nervous system? And also because the peripheral nervous system is invertebrate. Not working very well. Okay. Yeah. So maybe I use this. Yeah. Okay. Thank you. So if we look at the peripheral nerve system in vertebrates, the vertebrate peripheral nerve system developed mainly from two embryonic structures I just told you about. One is the neural crest, and the other one is the neurogenic placos. And both structures are developed from at the edges of the neural plate. And this, I just use this diagram to summarize the developmental mechanism or the process of neural crest and placos. And during the neurulation of vertebrae, if we make a cross-section of the dorsal ectoderm, we can see that in the most dorsal part of the dorsal ectoderm, you will start to form the neural tube. It curls up to form the neural tube. And the edges of the neural plate and the epidermal ectoderm, there will be a population of the cell that eventually fuse together at the most dorsal part of the neural tube. And then after that, those cells will become mesenchymal-like cells and elongate and migrate to their final destination and differentiate into many different kind of neuronal cell, glial cell, or even neuroendocrine cells. And even some of those cells will contribute to the craniofacial structure of vertebrate animal. So many of the anterior elaborate structure of our vertebrate head actually come from neurocrest cells. And during the past 20 years of research, people have already known that the vertebrate neurocrest need to be the development of the neurocrest requires a lot of this interaction of signaling molecule and transcription factors, and the interaction of those signaling molecule and transcription factors defines the border region of the neuroectoderm, and also confers the ability of those cells to become mesenchymal cell and differentiate into many different kinds of cell types. And turn off. And in another group of the embryonic tissue that I just talked about, the cranial placal cells, they come from ectoderm cells, and those ectodermal tissue will, during development, will become thickened, and some of those cells will start to become invaginate and migrate also into many places of the vertebrate body and differentiate into sensory neurons or lens structure. So many of the sensory organs in the vertebrates actually come from neurogenic placals, like the hair cells in our ear, and also the olfactory sensory neuron in our nose, or the lateral line sensory cells in the fish body. So if we look at many of the novel structure or characters of vertebrates are actually from neurocrest and placals. So these two, so this has been hypothesized that the evolutionary origin of neurocrest and partly from placals probably is the one of the pivotal factors that enable vertebrae to radiate into so many different kinds of body plans, and also enable them to become the top predator in our planets. So the original vertebrate novel structure, especially the neurocrest and placal, has been a question of our interest. So when we compare the developmental mechanism of cephalochordate amphioxus, we found that amphioxus also do the neurulation, just very similar to what I told you about in the vertebrate situation. Their neural tube, the neural plate also curl up during the neural stage and form the dorsal neural tube at the midline of dorsal midline of this animal. But we never found denominated cells from the dorsal part of the neural tube. So essentially, amphioxus, if we look at the morphology of this animal, its head region is relatively simple compared to the vertebrate situation. It does not have a very prominent eye structure, and it does not have like nose or ear. So essentially, there's no neurocrest derived structure or sensory organs around the amphioxus head. So it has been hypothesized this structure on the vertebrate novelties. But if we trace, but as we can say one phrase, nothing is come from nothing. There must be something to be worked on in order to give some evolutionary novelty. So when I started my my research on amphioxus, the evolutionary origin of neurocrest has been one of my main target interests. So when the amphioxus genome became available at 2008, I set up a comparative approach, try to identify how the neurocrest regulatory network can be evolved. So the first step is to identify the genes that have known involved in the neural crest evolution, to identify whether they are also present in the cephalochordate amphioxus genome. So when we do this whole genome comparison, we found that almost all the important regulatory genes, including the signaling molecules and transcription factors, are all present in amphioxus. They are all present, just less copy. So later I will talk about gene duplication. So the question becomes, okay, if all these genes are present, how come in vertebrate they can evolve neural crest and in cephalochordate they cannot? So I decided to compare the gene usage, the embryonic expression of all these genes in order to understand the origin of the neural crest gene regulatory network. So during this comparison, we found that the upper part of this network, the signaling molecules involving like BMP, Wnt, and FGF, they're all expressed in very similar way between amphioxus and cephalochordate and vertebrates. And also, the second part of this network, the transcription factors that defines the neural borders and the epidermis and the neural plate are also very similar. They are expressed in very similar way. For example, this SOX-B1 gene, as I told you, is a neural marker, and the dystolase gene marks the border of the neural plate. And this is in the situation, this has been shown in vertebrate situation that's very important to setting up the border of the neural plate. And we found that this is a dorsal view of the amphioxus embryo. It also labels, SOX-B1 is also labels the neural plate. Dystolase is also labels the border of the neural plate. And more importantly, the signaling molecules that control this border is also very conserved between amphioxus and vertebrates. In vertebrate, it has been shown that the BMP signaling level is very important to control this border. And in amphioxus, we demonstrate that when we manipulate the BMP signaling level, you can change the border of this dystolase expression pattern at the expense of the SOX-B1 neural expression. So this is so we demonstrate that this upper two level of the network is a very conserved genetic network for chordates to define the neural and non-neural border in the dorsal ectoderm. But we found that although amphioxus also have some transcription factors that are very important in the vertebrate lineage to define the neural crest cells. In amphioxus, those genes are not expressed at the border of the neural crest. Instead, they are mostly expressed in the mesodermal structure here, not in the border of the neural plate. So it suggests that in the vertebrate lineage, probably the intercalation of those genes into these genetic networks enable the border cells to gain some new cellular properties like denomination and migration. And we also compare some of the downstream genes that specify the differentiation of some neural crest cell to differentiate into different cell type. And we found that in amphioxus, those genes are also playing quite similar function, but somehow those cells cannot migrate. For example, MIF gene is a transcription factor that is control the expression of tyrosinase or tyrosinase protein that make melanocytes. And pigment cell melanocyte is one of the neural crest derivatives in vertebrate animal. In vertebrates, those cells will migrate out from the dorsal side of the neural tube and differentiate into pigment cells or melanocytes. But in amphioxus, those cells will retain inside the neural tube throughout their life, and they develop into some pigment cells. And we know we know that these pigment cells are probably responsible for the light sensing function in amphioxus. So it appears that generating pigment cells from neural tube is a very ancient trait for chordates, or even for deuterostome, for deuterostome or other animal. But somehow in the vertebrate lineage, those cells can acquire the ability to migrate out from neural tube and differentiate into more diverse cell types. So from those comparisons, we now know that the upper part of this genetic network is very conserved for setting up the neural borders, and also the downstream part of this genetic network, some part is also very conserved. But most distinct part is the central part of this network that specify the neural crest cell type and control the differentiation of neural crest. Although they are present in amphioxus, but they are not expressed at the border of amphioxus neural plate. So we hypothesized that in the vertebrate lineage, the initial step in generating the real neural crest cells is by the interculation of this part of the network into this platform under the control of the neural plate border. So how can we test this hypothesis? So we decided to focus on one of the genes, which is called the FOXD3. Is in this category of the cells. We know that it has been known that this gene is specifically expressed in the neural plate border, and its expression can control the adhesion molecule, the expression of adhesion molecule genes like E-cadherin E-cadherin. So and we also found this gene has undergone duplication and give rise to at least 5 paralogs in the vertebrate lineage. In amphioxus, we only found one representative of this gene family that we call the amphioxus FOXD. But in vertebrate, this gene have undergone gene duplication, probably due to the 2-round whole genome duplication, give rise to 5 genes. And we also compared the expression pattern of amphioxus FOXD and vertebrate FOXD family paralogs. And we found a very interesting phenomena that has been described called the subfunctionalization. That here I just use this cartoon to illustrate what had happened. So in amphioxus, we found this gene is mainly expressed in 3 major domains. One is in the forebrain, somites, and the other one is in the notochord. But when we compare the expression pattern of the FOXD paralog, we found FOXD1 paralog is expressed in the forebrain, D2 is expressed in the somites, and D4 is expressed in the notochord, and D5 is expressed in the somite and forebrain. And when we look at the FOXD3 expression, this gene is involved in the neural crest development, and it is specifically expressed in the border of the neural plate, and eventually they will become neural crest. So we start to make a hypothesis. We think if we use the amphioxus situation to represent an ancestral condition, we hypothesize that probably after gene duplication, the cis-regulatory element controls those gene expressions start to diverge and maybe acquire some complementary degeneration, and that results in the subfunctionalization of D1, D2, D4, and D5 into several different domains. And at the same time, FOXD3 probably acquire a new cis-regulatory domain that enables this gene to be expressed at the border of the neural crest. And we try to test this hypothesis by generating, by analyzing the cis regulation between amphioxus and vertebrae, and also doing some species experiments to test that. And we identify a cis regulatory element in amphioxus that can drive reporter gene expression that recapitulate the expression pattern in these three major domains. So we use this cis regulatory element and hook up to a EGFP reporter protein, reporter gene, and send it into a chick embryo to see what kind of expression pattern it can drive. And we found that this reporter gene can drive expression in the sow mice. So the green is the reporter gene, and the red one is the co-injected, just the tracing RFP construct. So we can see that this construct can drive expression in the sow mice, in the nodal core, and also in the neural tube. But they are not expressed in the neural border and not expressed in the migrating neural crest. That's suggesting that the amphioxus cis regulatory module do not have the required cis regulatory element to drive expression at the neural crest precursor. So that suggests this model might be correct, that during evolution, FOXD3 or FOXD gene family, this gene parallel evolve a new cis regulatory element, enable this gene to be expressed at the border of neural crest, and that co-option probably enable the cell to acquire some new cellular properties. Okay, so those data suggest that the regulation of FOXD3 paralog have changed during evolution. So we also want to address another issue, that whether during this, after the gene duplication, the coding sequence itself also change, and also change the protein function of those parallel genes. So we do some comparative study using in vitro and in vivo system to study the transcription, the regulatory activity of the amphioxus FOXD protein and other FOXD parallel from vertebrate lineage, and using gel shift mobility experiment, we found that the amphioxus FOXD protein can bind very similar binding sites compared to the Xenopus FOXD1, D3, and also to other FOXD parallel. And also, when we make a reporter construct that tests the regulatory activity of the FOXD parallel and from vertebrae and amphioxus, we found they are all working as transcription repressors. So biochemically, they have very similar function, recognizing similar DNA sequence and acting as repressor. But when we use in vivo system and overexpress those FOXD parallel or amphioxus FOXD gene, FOXD protein into a chick embryo, we found some quite interesting results. So here I just show you using a chick model to test the function of FOXD protein. And we found that the FOXD3 protein from Xenopus or zebrafish, they can both induce the ectopic expression of neural crest markers like SOX10 or HNK1 epitope positive cells at the injected site, suggesting they have the ability to induce ectopic formation of the neural crest. But when we test the activity of other FOXD parallel, like FOXD1 or D5, or the amphioxus FOXD protein, none of them can induce ectopic expression of the neural crest markers, suggesting that after gene duplication, those parallel have diverged their function in their coding sequence. And when we do when we do some domain shuffling experiment, and we start to identify, we want to pinpoint which protein domain is responsible for this change of the function in the FOXD paralog. And we found only a 39 amino acid at the very N-terminal region of the FOXD protein can confer the neural crest generating activity to amphioxus FOXD protein. And this protein domain is very conserved between all the FOXD paralog in different vertebrate animals, ranging from mouse, chicken, xenopus, zebrafish. That suggests that during the early evolution of vertebrate lineage, this FOXD3 somehow acquired a new protein domain that enabled them to interact with the other gene and create the neural crest formation. So in summary of this part of the talk, through the comparison of whole genome data and also the expression survey and also the transspecies experiment on their regulation and the protein function, we start to understand how the neural crest, the initial neural crest gene regulatory network might have evolved. We think from our data, it suggests that the assembly of this genetic network first involve the intercalation or co-option of this part of the gene, the neural crest specifier genes, into this network. And this new expression domain or the new expression of this group of genes enable those cells can confer those cells some new cellular property, including like the migration and differentiation into many neural crest-derived cell types. And we also demonstrate that after the duplication, some of those transcription factors might have already acquired some new function in their protein function. So both cis-regulatory change and coding sequence evolution contribute to the evolution of neural crest in the vertebrate lineage. So now I will shift my topic a little bit to the peripheral nerve system. So I just tell you that the evolution of neural crest probably is a vertebrate novelty, but amphioxus do have peripheral nerve system. So the question becomes whether what kind of homology or what kind of relationships between the peripheral nerve system of amphioxus to other vertebrate systems. So when we compare the peripheral nerve system with amphioxus, we found they are quite different from the vertebrate situation. Most of the amphioxus peripheral nerve system are primary sensory neurons. They are located, the sensory neurons are located outside the central nerve system, and they innervate directly into the central nerve system in amphioxus. And usually they do not have glia-like supporting cells, and usually and also they are not cluster or become sensory organs like in this vertebrate situation. So in order to understand the evolutionary relationship between those sensory neurons to the vertebrae or vertebrate sensory organs, we try to understand how these sensory neurons develop in amphioxus. So we have been focusing on one of the cell types that called the type 1 receptor cells. So these type 1 receptor cells are solitary sensory cells that are located on the epidermis of amphioxus. And on the apical side, they have a ring of shorter cilia, a microvilli, and in the central part, apical central part, it has a central cilium, single one central cilium. And during development, it starts to show at the peripheral of the epidermis, and using EM, we can see quite a lot of this kind of primary sensory neurons located on the epidermis. And it has been shown that these sensory neurons are derived from the ventral epidermis of the embryo, and during development, they will migrate upward from the most ventral part of the developing embryo and migrate in between the epidermis and the mesentery. And eventually, when they reach their final destination, they will re-erupt to the epidermis and send out cilium to the outside, and then send out axon to connect to the central nervous system. So they are primary sensory neurons. And in order to understand their development, so first we use a perneural marker, Hu-ELOF, to trace the development of those cells. So we can see that from the early neural stage, on the ventral side of those cells, of those embryo, we can start to see those spotty expression of hue that indicates the postmitotic sensory neurons located in this position, and eventually they distribute more dorsally, as I just explained to you. And we also noticed that a very important signaling molecule called delta is also expressed in very similar way, but its expression is prior to the presence of the Hu-ELOF expression. So we hypothesize probably the delta notch signal is very important for specifying those cells. So we use an inhibitor called DAPT to manipulate, disturb the delta notch signaling pathway. And we see that the distribution of those neuronal cells are affected by this manipulation. Delta notch signaling is working on specifying the single sensory neuronal cell from the epidermis. When we block this signal, we start to see clustering of the sensory cells from the ventral part of those embryo. And this is very conserved to the situation that has been found in other animals. And we also found that Archish-skewed transcription factor is working with delta notch signaling to specify those single neuronal cells in amphioxus. And when we look at, and also we notice that these cells are always present from the ventral part of the epidermis. And I have told you that the ventral part in the amphioxus embryo actually is the high BMP signaling part inside the embryo. and so we hypothesize probably BMP signaling level is important for specifying the epidermal sensory cell. And when we manipulate the BMP signal, we found that indeed, as I told you, that if we elevated the BMP signaling level, the central nerve system will disappear. But on the other hand, the distribution of those epidermal sensory cells will become all over the entire epidermis, suggesting that the specification of those sensory neuron is an independent event aside from the central nerve system specification. We further test that idea by manipulating the BMP signaling after the central nerve system is already established. And we show that indeed, even the central nerve system in those BMP manipulated embryo is not affected mostly, but if we wait until the central nerve system is specified and then manipulate the BMP signal level, we can either expand or reduce the formation of the central nerve system by either overexpress BMP or downregulate BMP signaling in those embryos. So that suggests that the epidermal sensory neuron patterning is an independent event that occurs later than the central nerve system induction in amphioxus. And currently, we further use different kind of genetic markers, developmental markers, to investigate the distribution of those sensory neurons, and we start to find that actually there are several subgroups of the sensory neurons in amphioxus, and they express different kind of markers during development. So we can roughly divide them into the ventral group of the sensory neuron that express this group of the gene, and later on there's another lateral part of the lateral group of the sensory neuron that's expressing different transcription factor and marker genes. So in comparison to the situation in other organisms, as I told you that most of the vertebrate peripheral nerve system is developed from either neural crests or placos, and placos are mostly derived from the epidermal ectoderm, but those epidermal neurogenic placos are mostly associated with in very adjacent position around the neural tube. But if we look at the sensory neuron development, not only in amphioxus but also in other organisms, including like cnidarian or even or in protostome, we can see that using the BHLH transcription factor and delta notch signaling to specify central nerves, to specify peripheral sensory neuron, is a very conserved mechanism to make sensory neuron. But somehow in the vertebrate lineage, they seem to centralize this mechanism or the domain of making sensory neurons to a more centralized region around the neural tube. So we hypothesized that maybe somehow during evolution, vertebrae evolved a way to not only centralize their central nervous system, they also centralized the domain that forms peripheral sensory neurons in vertebrate animals. Okay. Okay, I have talked about how this elaboration of the head and sensory system can be evolved in vertebrae. And in the last part, I will share with you some recent results that related to the cartilage and bone evolution in the chordate lineage. So, cartilage and bone are one of the definitive character in vertebrates, and because of those structure, vertebrae can have a higher ability of locomotion and also have a much stronger supporting ability to their body. But if we compare the connective tissues and also the supporting tissue in other invertebrate chordates, we found that although amphioxus or cephalochordate or urochordates, they do not have highly minorized skeletal tissue. They do have some supporting tissues that might be related to the minorized skeletal tissue in vertebrates. So here I just show you some of the morphological character in amphioxus that has been suggested, or we think there might be the evolutionary precursor of vertebrate skeletal tissue. For example, in the anterior part of amphioxus, they have this oral series. They are collagen-based structure that shows the mouth opening to keep the big particle out of their incurrence flow for feeding. And also within the pharyngeal region, we can also notice there are some collagen-based gill-slit structures, and that's in here, that's supporting the pharyngeal region. And also underneath the epidermis, we can observe some collagen-based tissues underneath the epidermis, and those collagen structures also provide some supporting abilities to this animal. But compared to the vertebrate situation, those structures are not minorized, so they are not really like skeleton. But we want to know what kind of mechanism or genetic mechanism, developmental mechanism that enables amphioxus to develop those kind of structure, and hopefully using those knowledge we can start to understand whether they are homologous to the vertebrate skeletal tissues. And we collaborate with our colleague in University of Colorado, our colleague Dan Mullerman-Menderos in his group, to study how the oral tentacle, this oral series, develop. And also we compare the cellular structures to the vertebrate gill bars. And quite interestingly, we found that the amphioxus oral skeleton and those oral tentacles are very similar to the gill bar cartilage in terms of their cellular morphology and also their extracellular matrix, and also the cell arrangement. And also we compared the genes that have been known important for the development of cellular cartilage in vertebrae, and we found the homologous gene in amphioxus is also expressed in the primordial of those cellular of their oral series. For example, the SOX-E, SOX-D transcription factors are very important for the cellular cartilage development in amphioxus, and also the collagen is a very important structural protein for cartilage. And we found that they are all expressed in the primordial of the oral series. And another study also demonstrate that other cellular structure, other cellular extracellular matrix proteins are also important for the regeneration of those oral series in amphioxus. That further support the idea that this oral series might be related to the cellular cartilage in vertebrates. But question remains, we still don't know what is the embryonic origin of those, we call it the cartilage-forming cells in amphioxus. So we try to understand this by looking at, by tracing the expression of those extracellular protein genes to have a better understanding of this question. So we start to focus on this question by using one of the extracellular protein, SPARC, as a marker to look at it. And this SPARC gene is no exception during the vertebrate evolution. It also duplicates many times to give rise to a big group of extracellular proteins. And during the evolution, this SPARC gene first duplicate to give rise to a SPARC-L1 gene, and this SPARC-L1 gene further tandem duplicate to give rise to a group of genes called SCPP genes that are very important for the dentin animal, the minorized tissue formation. And when we look at the expression pattern of this gene, we found that it starts out, is expressed at the lateral part of the amphioxus somi. As you can see that only with the progress of the development, only the lateral part of the somi is expressed this gene. And also during development, this lateral part of the somi will grow downward ventrally to surround the entire animal, and also to surround the neural tube and also the guts. And also this lateral part of the somi is different from the medial part of the somi that will eventually contribute to the myomere of the amphioxus. And we can use like the muscle acting expression to differentiate the different domain of amphioxus somi. And we also do in situ of other extracellular proteins, and they show very similar co-expression pattern with the spark protein at the lateral part of the somi, and eventually covers the neural tube and also the gut. And later on, when we do gene expression survey in the juvenile, we found that the spark expression positive cell are associated with the cell that encompass those collagen structures I just showed you before, and also it is in the oral series primordial in those juveniles. And from the EM, we can see quite clearly that those spark positive cells seems to associate with the collagenous mesh structure underneath, besides the neural tube, and also besides the nodal core, and also underneath the epidermis. And from the knowledge of skeletogenesis in vertebrae, it has been known that most of the skeletogenic cells in vertebrae come from the skeleton and also the dermomyotome that developed from the vertebrate somites. So from the observation in amphioxus, we also found those extracellular proteins are expressed at the lateral part of the amphioxus somi. So we are wondering whether in amphioxus there are similar compartments of the of somi compartments compared to the vertebrate situation that can contribute to those collagen forming tissues. So we start to use multicolor in situ hybridization that employ that using the marker genes that have been known that can mark different parts of the vertebrate somites, and we map them, the expression pattern of those genes, into amphioxus developing somi, and we start to see very clear compartmentalization in the amphioxus developing somi. So now we can recognize there are at least one medial domain in the amphioxus somi that usually express myotomal markers, and there are three. Now we can recognize three lateral domains that express different kind of transcription factors that mark different part of those domains. But one of the interesting things we found is most of the skeletal markers that have been reported in vertebrates, their homologs are not expressed at the somites. That indicates probably the amphioxus somi do not really have the full genetic repertoire for forming the skeleton, but they seems to have other genetic components that can make dermomyotome or lateral plate mesoderm parts. We further test this hypothesis by investigating how these skeleton, these somi compartments being specified in amphioxus, using the knowledge that has been studied in vertebrates to test their homology. One of the very important signaling pathway that can affect the somi lateral patterning, lateral and medial patterning, is the BMP signaling level. And we also see that in the developing amphioxus somites, the BMP signaling level seems to be much higher at the lateral part of the developing somi compared to the medial part of the somi. Suggests maybe BMP is involved in patterning the lateral and medial compartments in amphioxus. And we also did some manipulations, either inhibit BMP signaling by inhibitors or elevated BMP signal by recombinant protein. And we did see some changes of the medial and the lateral compartments, depends on you block BMP or elevated BMP. And we also use different region-specific markers to further look at these changes of domain, and we can see that when we downregulate BMP signaling by the inhibitor, the lateral markers are usually shrinked. And on the other hand, when we overexpress BMP or elevating BMP signaling level by recombinant protein, we can get an expansion of those lateral markers, suggesting the BMP signal level indeed controls the lateral medial patterning of the somite compartments in amphioxus. So from those comparisons, we conclude that probably at the common ancestors of vertebrates and amphioxus, the chordate ancestor, the compartmentalized somi already exist. That has been our data strongly suggests that this compartmentalized somi has already exist, and the lateral part of the somi probably can contribute to the formation of supporting tissues in amphioxus. And also, during the evolution of vertebrates, the duplication of this spark gene family probably enables those protein family to evolve a highly minorized ability to further strengthen those protoskeletal tissue into highly minorized skeletal tissue in vertebrates. So we think that the duplications of spark gene and their neurofunctionalization of these parallels may have initiated intensive minorization of vertebrate skeletal tissues in vertebrates. Yeah. Okay. So I just conclude my talk with these slides. I hope today I convince you that amphioxus is a good system to study the common developmental mechanism of chordates, and also the study of amphioxus can give very good insights into how this vertebrate nervous system evolve. So we think using amphioxus, we can, and also the comparative study between amphioxus and vertebrate, we can generate some testable hypotheses and also design some interspecies experiment to explain how the vertebrate novel characters can be evolved during. So at the end, I would like to thank the people in my lab who actually did the work, including many of my former postdocs and students, and also technicians. They helped to do the actual work on those projects, and also maintaining the animal resources and culturing in my lab. And I would also like to acknowledge many of the researchers along my research career helping me on those amphioxus research. And at the end, thank you for your listening.
Thank you very much. Any questions?
Concerning the OX-D3 let's say novel cis-regulatory element, do you have any clue on what this could be?
Yeah, I think that's a very important question. Now we are hoping to identify that. Just several years ago, Marianne's lab, they identified the vertebrate FOXD homolog, FOXD cis-regulatory element that enables this gene to be expressed at the border of the neural crest. So right now, we have prepared some of the reporter constructs. So hopefully we can win them back and inject into amphioxus to see what kind of expression pattern they can generate. So we hope also by this kind of back experiment can help us to understand, pinpoint the cis-regulatory element that enables this gene to be expressed in the vertebrate image. Yeah.
Are there any information on the symptomatic use of
Pigments
Yeah.
mostly I think is melanin. Yeah, the black. Yeah. Yeah, that's melanin. Yeah.
Thanks a lot.
Okay, thank you.